Okay, now we're going to cover Chapter 14, Section 2, Human Genetic Disorders. Our key questions are, how do small changes in DNA molecules affect human traits? And what are the effects of errors in meiosis? Roman numeral 1 in our notes, from molecule to phenotype. Number 1, there is a direct link between genotype and phenotype. Genotype is the genetic makeup, the genes that are present, the genes that are passed down to us from our parents. And phenotype is the physical traits that we see, the outcome of the genes. An example here is earwax. Earwax has two different forms, wet or dry. People of African and European ancestry are more likely to have wet earwax, the dominant form. Those of Asian or Native American ancestry most often have a dry form, which is recessive. A single DNA base in the gene for a membrane transport protein is the culprit. A simple base change from guanine to adenine causes this protein to produce dry earwax instead of wet earwax. Number 2, how do small changes in DNA molecules affect human traits? Changes in a gene's DNA sequence can change proteins by altering their amino acid sequences, which may directly affect one's phenotype. In other words, there is a molecular basis for genetic disorders. There are thousands of genetic disorders that are caused by changes in individual genes. These changes often affect specific proteins associated with important cellular functions. Number 3, sickle cell disease. The main cause of this disorder is a defective allele for beta globin, one of two polypeptides in hemoglobin. Hemoglobin is the oxygen-carrying protein in red blood cells. The result of sickle cell disease are sickle-shaped cells, red blood cells, that are more rigid than a normal red blood cell, and therefore they tend to get stuck in the capillaries. The blood stops moving through the capillaries. This can cause damage to cells, tissues, and even organs. Cystic fibrosis, also known as CF for short. It's most common among people of European ancestry. The cause of cystic fibrosis is the deletion of just 3 bases in the gene for a protein. The symptoms of cystic fibrosis. 2 copies of the defective allele are needed to produce the disorder, which means that it is recessive. The symptoms are serious digestive problems that produce a thick, heavy mucus that clogs the lungs and breathing passageways. Huntington's disease. This is caused by a dominant allele for a protein found in brain cells. The allele for this disease contains a long string of bases in which the codon CAG that codes for an amino acid glutamine repeats over and over and over again, more than 40 times. Despite intensive studies, the reason why these long strings of glutamine cause disease is still not clear. The symptoms of Huntington's disease, namely mental deterioration and uncontrollable movements, usually do not appear until middle age. The greater the number of codon repeats, the earlier the disease appears, and the more severe its symptoms are. Genetic advantages. The cystic fibrosis, or CF allele, helps protect against typhoid fever. More than 1,000 years ago, the cities of medieval Europe were ravaged by epidemics of typhoid fever. Typhoid is caused by a bacterium that enters the body through cells in the digestive system. The protein produced by CF allele helps block the entry of this bacterium. Individuals that are heterozygous for cystic fibrosis would have had an advantage when living in cities with poor sanitation and polluted water. And because they also carried a normal allele, these individuals did not suffer from cystic fibrosis. Sickle cell allele also helps to resist against the parasite that spreads malaria. Most African Americans today are descended from populations that originally lived in West Central Africa, where malaria is common. Malaria is a mosquito-borne infection caused by a parasite that lives inside red blood cells. Individuals with just one copy of the sickle cell allele are generally healthy and are also highly resistant to the parasite. This resistance gives them a great advantage against malaria, which even today claims more than 1 million lives every year. We take a look at the picture. Okay, in purple, it is highlighting the areas where malaria is common. In yellow, it is highlighting the areas where sickle cell allele is common. And in this last picture, it's showing the overlap between the sickle cell allele and malaria. Chromosomal disorders. Number 1, non-disjunction. Non-disjunction is an error in meiosis in which the homologous chromosomes fail to separate properly. It literally means not coming apart. If we take a look at the video here, okay, starting at the top, we have 2 red chromosomes and 2 yellow chromosomes inside the cell. Non-disjunction occurs during meiosis 1, which leaves us with 1 red and 2 yellow in the cell on the left and only 1 red in the cell on the right. If it was going to occur correctly, if meiosis 1 would have occurred correctly, we should have 1 red and 1 yellow in each of those 2 cells. Meiosis 2 occurs, and it's leaving us with 4 cells. The first one, a red and 2 yellow. The second one, a red and 2 yellow. And the third and fourth, just with a red. If meiosis occurred correctly, we'd have 1 red and yellow in each of the 4 cells. Non-disjunction results in trisomy, which means that there are 3 copies of a chromosome in some of the cells. Trisomy can result in Down syndrome. The most common form of trisomy involving 3 copies of chromosome 21 is Down syndrome, which is often characterized by mild to severe mental retardation and a high frequency of certain birth defects. Two other examples of non-disjunction occur in male and a female. Non-disjunction of the X chromosomes can lead to a disorder known as Turner syndrome. A female with Turner syndrome usually inherits only 1 X chromosome. Women with Turner syndrome are sterile, which means that they are unable to reproduce. Their sex organs do not develop properly at puberty. This is the cell on the right side of the picture. In males, non-disjunction may cause Klinefelter syndrome, resulting from the inheritance of an extra X chromosome. So since males are usually XY, this would result in XXY, which interferes with the meiosis and usually prevents these individuals from reproducing. There have been no reported instances of babies being born without an X chromosome, indicating that this chromosome contains genes that are vital for the survival and development of the embryo. And that wraps up Chapter 14, Section 2.